Good morning, good afternoon, or good evening, everyone. On behalf of Microtracers, it is my absolute pleasure to welcome you all to today's webinar. And a special thank you to Antonio and Rita. We are deeply honored by your participation and support. And we are delighted to see so many of you here from diverse backgrounds and with unique perspectives. And we are sponsoring this webinar to educate the general public, as well as pharmaceutical and regulatory authorities on the novel on-dose authentication technology, SECURtracers, and how it can be used to code solid oral dosage form drugs. And as a team, we will provide the background of microtracers and speakers, address the scope and impact of counterfeit pharmaceuticals, evaluate current anti-counterfeiting measures and their limitations, discuss PCIDs and the regulatory hurdles for implementation, and explore SECURtracers and its potential to effectively combat counterfeit drugs. We will have the opportunity today to exchange ideas, explore collaborative initiatives, and deepen our understanding of the challenges and opportunities that lie ahead. And I'm confident that the insights shared today will be both enlightening and actionable. And so before we begin, I'd like to take a moment to introduce today's speakers. So Zachary Eisenberg has worked for Microtracers since 2011. He is the son of current president David Eisenberg and the grandson of founder Dr. Sylvan Eisenberg. And after graduating with his MBA from the University of Michigan Ross School of Business in 2015, he has acted as vice president and COO of both Microtracers and our affiliated analytical and RESCO laboratories. And so next, I would like to invite Dr. Nikolay Barashkov to introduce himself as well.
I am Dr. Nikolay Barashkov. I hold dual PhDs in physical chemistry and polymer chemistry and have about 40 years of experience in polymer chemistry, photochemistry, photophysics, and nanotechnology. I have co-written 8 books, more than 150 peer-review papers, and hold 30 patents. As the Director of Research and Development and Technical Services at Microtracers, I am responsible for R&D and QC for all microtracers, including newly developed SECURtracers.
And next, I would like to open the floor to Stephen Keteltas to introduce himself as well.
Hi there, my name's Stephen Keteltas. I am the lead manufacturing chemist here at Microtracers. I graduated with a bachelor's degree in chemistry and biochemistry from Florida State University, and I then went on to obtain my master's degree in chemistry from the University of California at San Diego. Since 2015, so about a decade now, I've been working here at Microtracers as the lead manufacturing chemist on SECURtracers, the product that we're going to be telling you about today. And I also work on various other research projects here for the company.
And lastly, I'd like to introduce myself as well. I'm Alyssa Tabucao. I earned my Bachelor's of Sciences degree in medicinal and synthetic chemistry from the University of San Francisco. I serve as the product manager for SECURtracers product. I typically oversee the marketing, development, and improvement of SECURtracers, as well as ensuring compliance with XCPACT, GMP, and ISO regulations. And I will be guiding today's discussion. And now I will turn it over to Zach again to speak about the history of Microtracers.
So Microtracers was founded by Dr. Sylvan Eisenberg, who's my grandfather, in 1961, as a means of providing, as a rapid means of identifying drug products found in animal feeds. So our simplest microtracers consist of iron grit particles coated in food dyes. When formulated into a drug or high-value ingredient, they can be retrieved from premixes or final feeds magnetically, with the retrieved material put onto a filter paper and sprayed with a developing solution. By the color of the spots, you can identify what drugs are present in the feed or a suspect sample. By the quantity of the spots, you could estimate the quantity of that drug in the sample. So it's a very simple solution, but it has very profound use cases. In addition to coding feed products as proprietary, which is the primary reason, it's also used to determine, to validate mixing and contamination control procedures. So microtracers are now sold all over the world. We have distributors in over 25 countries, and some of our major customers include Elanco, which is formerly part of Eli Lilly, Zoetis, which is formerly part of Pfizer, Phibro Animal Health, and many more. In total, we estimate that our tracers have been formulated into about 2 billion tons of feed without a single adverse reaction reported, 200 million of which within the last year or so. And then a little bit about the background of SECURtracers. Because we are limited by the number of carriers and colors available, we've been somewhat victims of our own success. So we're always looking for new tracers that could be applied for our use case in animal feeds. One idea that my grandfather had was to microengrave particles rather than coat them in food dyes. And then these could be read via an inexpensive microscope. He ran a patent search in 1997. I think he also tried to make some prototypes, but the technology just didn't exist at that time. It's only more recently that like the world has kind of caught up to the idea and made it feasible again. So in 2013, we had my father, David Eisenberg, met with Lou Kish, who's a pioneer in the semiconductor industry, working at Fairfield Semiconductors, I believe, in Silicon Valley, with some of the very first individuals who ended up founding Intel and some of these other companies. In any event, he was advising that it would be feasible to manufacture these tracers using semiconductor technology, and that they had the precision and the capability to manufacture particles that are consistent and have such writing on each of them. So with the help of then Dr. Jonathan Germain, we were able to actually develop this capability, and we call it SECURtracers.
And so, leveraging decades of our parent company's expertise, we believe that SECURtracers are now positioned to facilitate this paradigm shift in human drug authentication. And I'd like to shift focus to the issue of pharmaceutical counterfeiting as well as its impact. So, as you may all be aware, pharmaceutical counterfeiting is a critical global health issue that threatens patient safety. And according to the Pharmaceutical Security Institute, in the year of 2023, there were nearly 7,000 pharmaceutical crime incidents, marking a 4% increase from 2022 and a 15% increase from 2021, as you can see here. And the counterfeit drug market generates a staggering $200 to $400 billion annually and is responsible for over 1 million preventable deaths each year. And counterfeiting is typically driven by numerous factors, but I just wanted to focus on the few that you can see on this slide here. So high cost of proprietary medications. So the steep price of legitimate drugs incentivizes counterfeiting as a lucrative criminal activity. As well, we've got increased internet sales. So online drug sales provide a fertile ground for counterfeiters with limited regulatory oversight in many regions. There's also minimal legal risks. So in many jurisdictions, the penalties for counterfeiting are not severe enough to deter such criminal activity. And lastly, ease of production. So advances in technology have made it easier for counterfeiters to produce convincing fake drugs, as you can see to the right. And counterfeiting thrives particularly in developing countries where traceability is limited and price sensitivity is high. So for example, in Africa and in Nigeria specifically, counterfeiters often target life-saving medications like antimalarials and AIDS treatments. And this is commonly facilitated by the practice of selling medications in open air markets, where pills are frequently sold loosely or in easily replicable packaging. And alarmingly, Africa accounts for nearly half of all global incidents of counterfeit medicines. However, the threat is not confined to these developing nations. In developed markets, high drug consumption combined with increasing reliance on online pharmacies exacerbate this problem. And the COVID-19 pandemic caused a surge in online pharmacy use, including unlicensed pharmacies. And the World Health Organization reported that 50% of online pharmacies concealing their physical addresses were found to be distributing counterfeit drugs. And as I mentioned previously, counterfeiters remain undeterred by the legal risks associated with their operations. So, for example, in California, a counterfeit drug offense can result in either a misdemeanor or a felony charge, depending on the circumstances. And the misdemeanor charges may lead up to a jail sentence of 6 months or a fine of up to $1,000. And now I would like to shift gears and have Zach talk about existing anti-counterfeiting measures to combat this global issue and discuss their limitations.
Great. Thanks, Alyssa. All right, so the first solution we're going to discuss is track and trace and serialization. So this is a system that's being implemented in the United States and the European Union. I know that many other countries are also considering implementing track and trace, and some are under the impression that it completely eliminates the potential for counterfeiting, and that's unfortunately just not the case. There are pros to the system. It provides real-time monitoring because these packages are being scanned at various different levels of the supply chain. That there's very good end-to-end visibility as long as those products are scanned and also present where they're supposed to be. So within the supply chain, there's very little potential for error there. But the cons are it's extremely expensive and difficult to implement. I think even in the US and EU, it's still not fully implemented, even after it was mandated to be so. And it's been estimated to add 8 to 10% to manufacturing costs, which is huge and certainly material. And there are some failures as well. So the effectiveness significantly diminishes outside of the supply chain. So once it gets to the pharmacist or the end consumer, it's no longer tracked, it's no longer traced. And those barcodes, those serial numbers can be easily produced and replicated and put onto packaging. And so you see that fraudulent QR codes and serial numbers are oftentimes included on counterfeit products, and verification is not really entirely possible after it's dispensed to the end user. So it has certain limitations when it comes to authentication. And then there's also various packaging-based solutions. So I'm sure people have seen holographic labels, tamper-proof labels that are sometimes applied to packaging. And the benefit is, the consumer has an immediate visual means of identifying products, and oftentimes these solutions are very inexpensive. The cons of these packaging-based solutions is that counterfeiters can oftentimes easily replicate them, whether they be RFID tags, holograms, tamper-proof labels, etc. And in some instances, there's been reports that the counterfeit holographic labels, for example, have looked more sophisticated than legitimate holographic labels. In which case, the consumer is not really able to easily differentiate which one is authentic and which one's not. So it requires a lot of education of the consumer, and even then, that might still not be enough to provide the assurance or 100% assurance that a product is authentic or not. And then in terms of failures, the packages are oftentimes reused or substituted or easily counterfeited, as we mentioned before. So just because a package looks like it might be authentic or it might even be authentic packaging. Doesn't mean the pharmaceuticals contained are authentic as well.
Great, thank you, Zach. I will now shift gears and let Stephen talk about PCIDs and their regulatory hurdles for implementation and where our product SECURtracers fits into this scheme.
All right, thanks, Alyssa. So here at Microtracers, we developed and we've been working for a while now on a new product called SECURtracers. So unlike the packaging-based solutions that Zach was just telling us about, SECURtracers are a novel authentication solution that actually goes on a solid dosage form itself. So it's a novel on-dose authentication solution, and it belongs to a class of anti-counterfeiting technology that's known as Physical-Chemical Identifiers, or also known as PCIDs. Our technology adheres to the 2011 FDA guidance for industry on incorporating PCIDs into solid oral dosage form drug products. According to this guidance, a physical-chemical identifier, or PCID, is defined as adding a trace amount of an inactive ingredient to authenticate legitimate drugs and to identify counterfeits. Common examples of PCIDs include inks, pigments, flavors, and molecular taggants. According to the FDA, PCIDs should be pharmacologically inert, ensuring that they don't affect the active drug's safety or efficacy, and PCIDs can be considered pharmaceutical excipients. The placement of the PCID is crucial for ensuring their safety and effectiveness. Integrating them into the core of the solid oral dosage form drugs could lead to interactions with the active pharmaceutical ingredient, potentially causing degradation of both the active pharmaceutical ingredient and the PCID or tracer. So therefore, placing the PCID in an external component of the formulation, such as the coating of a pill or a tablet, that's recommended to mitigate those risks, and as is, using the minimum level that's necessary for clear identification of the excipient and the medication. Additionally, the FDA recommended pharmaceutical manufacturers use direct food additives that are generally recognized as safe. This is commonly known as GRAS, G-R-A-S, and recommended using ingredients that are listed in the FDA's Inactive Ingredient Guide, also known as the IIG. PCIDs that meet these criteria can be incorporated into already approved drugs as a level 1 post-approval change. This offers the simplest pathway to implementation. But unfortunately, the criteria allowing for such a level 1 post-approval change are quite limiting. Other PCIDs, including our SECURtracers, require that a pharmaceutical manufacturer include the PCID in a new drug product submitted under a new drug application or an abbreviated new drug application. So these would be NDA or ANDA. Once the FDA has approved such a use, the FDA agency will consider including the tracers in the IIG, and that would make the post-approval route possible for subsequent users of the excipient. All right, I'll now transition to focus on our SECURtracers. Our on-dose authentication, SECURtracers, facilitates the identification of counterfeit drug products by absence of the tracer itself. So unlike the traditional packaging-based security measures that Zach was talking about, our on-dose authentication provides direct method of verification, and this remains with the pharmaceutical product itself throughout its lifetime, from the point of application to the point of end use. All right, so SECURtracers can be integrated into the coating of a drug, the matrix of a pill or tablet, or the shell or band seal of hard and soft capsules without adding any new equipment or processes to the manufacturing process. And here's a closer look at SECURtracers under a microscope. So you can see there, each one of the SECURtracer particles is pretty nice rectangular shape, like really nice, well-defined lines along the rectangle there. The dimensions are typically 200 microns in length, 100 microns in width, and about 10 microns tall. And these particles typically have a count of about 4 million. So those rectangles that you saw there, 4 million of those rectangles per gram. What you see here is a photo of the product in a glass vial, and it's pretty cool to see it even just with the naked eye. It looks almost kind of like glitter, and it's got this kind of nice golden, sort of greenish, golden yellow fluorescent color. Each particle on the microscopic level contains 10 or more microengraved letters or symbols. Both the dimensions of the rectangle itself, to some extent the shape, and also the microengraved pattern that you see, the letters that are etched in, those are customizable to meet customer requirements. On a more molecular scale, secure tracers are composed of entirely GRAS materials. So again, GRAS is generally regarded as safe. These materials are ethyl cellulose USP grade. That's the matrix, the ethyl cellulose matrix. Within this matrix are embedded curcumin. Curcumin is derived from turmeric oleoresin. Commonly, this is seen in kitchens. Any recipe that would include turmeric is going to have this curcumin in it. And like those dishes, it gives our product the kind of golden yellow, slightly greenish fluorescent aspect. So also embedded in the ethyl cell matrix is what we call carbonyl iron powder. Carbonyl in this case just refers to the purification method applied to making the iron pure. It's a very, very effective purification method yielding very high degree of purity of iron. Ethyl cellulose serves as what we call the matrix scaffolding of the tracer. While the functions of the curcumin and the carbonyl iron powder, those are critical to allow both secure tracer identification as well as secure tracer retrievability. Both of these things allow for rapid verification of drug authenticity when the particles are seen on the pharmaceutical dosage itself. More specifically, when secure tracers are integrated into a drug coating, curcumin exhibits the bright fluorescence that I was talking about. This occurs when shining a UV blacklight on the surface of a pill. It's around 360, 370 nanometers blacklight. This feature enables a very quick, very effective preliminary test. So just simply scanning the drug product surface with this UV blacklight is going to reveal the yellow gold fluorescent specks that you saw in the previous slide. Meanwhile, the absence of such specks, that would indicate a counterfeit product when the specks are not seen. Also notably, in normal ambient lighting that doesn't contain blacklight, the secure tracers are practically invisible, and that leaves the drug's appearance unchanged on the macroscopic level under ambient light. The carbonyl iron powder that I was talking about, this makes the tracers magnetically retrievable. So when the drug product is dissolved in water, the tracer particles can be isolated on a rare earth magnet in the lid of a small plastic jar. Then using a very simple, very inexpensive, readily available 60 to 120 time magnification handheld microscope, the microengraved letters that you saw on a previous slide, or symbols, whatever is etched into the rectangle, that can be read and determined to be a match with the tracer that's formulated for that specific drug product, thereby confirming the authenticity of that drug product. All right, now we are going to play a video illustrating our magnetic recovery procedure. All right, yeah, cool video. Thanks, Alyssa. Using this method, you can confidently authenticate your drug products without the need for costly detection devices, without the need for laboratory analyses, specialized training, or proprietary apps and databases. Our solution leverages low-cost tools like a pocket microscope, a UV flashlight, and a magnetic recovery jar, making it feasible for mass distribution to compliance teams, customs agents, drug enforcement officers, and other stakeholders. This enables very easy, on-the-spot verification across global markets, ensuring product integrity anywhere in the world. Typically, SECURtracers are added to solid oral drug products at a concentration of 12.5 to 25 ppm. This equates to 12.5 to 25 grams of SECURtracers per metric ton of drug product. Extensive testing using a variety of testing methods has shown no detectable impurities found, with a maximum level of detection of 250 parts per million. If the SECURtracer is formulated at 12.5 parts per million in the drug product, this level of detection would equate to a maximum for any identifiable impurity of only 3 parts per billion, 3 ppb, in the drug product itself. Both the US FDA and the European Medicines Agency have advised that such low levels of impurities as a maximum are not significant. This SECURtracer formulation is adequate. So the low formulation level that I just said, that's adequate to assure the presence of tracers very easily using the microscopic method, even if the mixing is not complete. For more cost-effective solutions, yet still, even smaller SECURtracers measuring only 50 by 50 by 10 microns, so small squares, containing 2 micro-engraved characters. Those particles can be used at concentrations as low as only 2.5 parts per million, while still adhering to FDA PCID guidelines and minimizing costs.
I'd just like to jump on here and mention that the FDA's recognition of PCIDs really emphasizes their role in enhancing drug traceability, which is what we at MicroTracers are really trying to do here. So over the past few years, we have made significant advancements that I'd like to highlight, but I'd only like to focus on a few here. So in January of 2022, we filed a drug master file, otherwise known as a DMF, with the US FDA. And for those who are unfamiliar, a DMF supports regulatory requirements and demonstrates the quality, safety, and efficacy of a medicinal product, and can allow parties such as pharmaceutical manufacturers to reference material in their drug application. In May of 2024, we at MicroTracers achieved ISO 9001 and 2015 accreditation, as well as AXIPACT GMP accreditation for pharmaceutical excipient manufacturing. And recently, we have partnered with the Bombay College of Pharmacy in Mumbai just to study the chemical change of SECURtracers. And we wanted to identify and determine the photodegradation of our products and the plasma degradation of curcumin through various methods. And through this, we just hope to determine if the chemical composition of the mixture of raw materials is changed throughout the manufacturing process. And lastly, we are conducting in vivo studies in collaboration with the Nigerian Institute of Medical Research to demonstrate the safety of SECURtracers as PCIDs in solid oral dosage form drugs. And our team has met with Nigeria's equivalent of the FDA, otherwise known as NAFDAC, to obtain regulatory feedback on the use of SECURtracers. And based on the recommendation, we plan to transition directly to a clinical trial following the in vivo studies, and we've already started working with the University Hospital, the NIMR, and NAFDAC to develop these clinical trial protocols. And so with this, we've got many more developments upcoming, and as of now, I'll just let Zach discuss the next steps and our future goals for SECURtracers.
Sure. So, as previously mentioned, I believe pharmaceutical manufacturers in the United States, at least, even though SECURtracers qualify as PCIDs, they do need to submit an NDA or ANDA to obtain regulatory approval for the addition of the tracers. In other markets, the regulatory requirements are different. So in Nigeria, such a process might not be required. But in any event, we're actively seeking pharmaceutical manufacturers to partner with us in integrating SECURtracers into solid oral dosage form drug products, regardless of the location. I think we were looking to develop test cases and to show that our products can be efficacious in authenticating products and also reducing the incidence of counterfeiting sensitive drugs. So to that end, for anybody that is willing to partner with us, we are willing to commit to a period of 3 years at providing the SECURtracers at no cost, providing detection apparatus, 200 or as many as may be necessary to provide it to whoever in the supply chain our customer believes best to receive them, whether that's their own anti-counterfeiting department, whether it's custom agents, pharmacists, whomever. We will provide these kits at no cost as well. We're also willing to compensate reasonable direct costs for registration changes, trials, and other commercialization requirements. And most importantly, we're looking to promote this usage too. So after the test case is developed, we will be promoting supporting the use of the SECURtracers to enhance the public awareness of the presence of the tracers and also to demonstrate that it was efficacious. So we hope that we'll find some people to join us in our mission to protect patients and uphold public health.